Okay, so may buntag, may hapon, may gabi sa tanan. Okay, so it doesn't matter what time of day you have the chance to look at this lecture video. So what's important is that you're able to uh, review the material for because we will not be able to meet as frequently dahil ang daming walang klase. Okay, so the learning objectives is, of course, for you to be able to discuss concepts and principles and significance of infection control and laboratory safety. Of course, we will also be tackling virus sterilization and disinfection methods for the first part. Okay, so again, reminder, this is a three-part lecture video. So the first part would be control of microbial or bacterial growth. Okay, so before we progress further into the discussion, so let's first define terminologies, diba? Ito yung mga words na lagi nating naririnig when we're talking about control of microbial growth. So number one, we have decontamination. So when we say decontamination, it refers to the removal of dirt or materials that would be on your on infectious organisms that would be on your materials na to be used for the laboratory. Now, it does not necessarily mean when you decontaminate, you'd be removing infectious materials. Okay, so it has to be. It could either be successful in removing in removing your pathogenic organisms, or it could just simply remove non-pathogenic organisms. Okay, so decontamination may be sterilization or through disinfection. Ayun, no? So pag nag-decontaminate using a process of sterilization, ayan, sure na you will be removing non-pathogenic as well as pathogenic organisms on the materials. Okay? Now, pag disinfection naman, this is the type of decontamination wherein you'll be removing only what is non-pathogenic or what is normally growing on the surface of your material, but it will not actually remove pathogenic organisms. Okay. Now, apart from these three major terms, you'll be coming across the words disinfectant. So these are antimicrobial agents that you can use to remove non-pathogenic organisms and some pathogenic organisms on inanimate objects that we come to use day in and day out. Antiseptic, on the other hand, are uh, antimicrobial agents that we use really safely on infected living tissues, no? Kagaya nito yung ating betadine, okay? So si alcohol, hindi antiseptic yun. Disinfectant lang. Now, the term degerming means to remove materials that may harbor germs, no? So pwedeng yung naghuhugas ka ng plato, uh, naglilinis kayo ng inyong CR, that's degerming. Okay, sanitation naman is a general term which we use to make sure that your living quarters, your environment is free from dirt. Okay, so nagsasanitize ka. Pwedeng yung maggamit ng gloves, no, para pag hindi nagta-transfer. Pwedeng yung kailangan malinis yung iyong mga garbage bins. Meron kang nakaseparate talaga na garbage bins. That's part of sanitation. Yung hindi pwedeng magtapon ng basura sa kanal, that's part of sanitation. Okay? Sepsis would mean the presence of infectious material sa ating sa body natin. Okay? Infectious material or the infectious agent plus their dead bodies and their toxins. No? So, sepsis yun. Present sila sa katawan natin. Now, the opposite of that would be asepsis, no? The absence of infectious material in our body. Okay, so ito yung target natin when we're working in the laboratory. There should always be asepsis or the absence of infectious or contaminating material. Kasi nga, di ba sa lab, ang ating gawain ay to identify the causative agent that is present in or that has caused the patient's ailments or disease, right? Now, there are several procedures that can help us acquire or achieve asepsis, and those procedures are called aseptic techniques. So aseptic techniques would include, number one, to make sure that everything is clean in your laboratory. So you use disinfectants, you use gloves, you use your lab gowns or your PPE. That's part of aseptic technique. You want to use new materials, okay? Or if you need to reuse it, then make sure to sterilize it first before using it in your assays. Okay. Then bacteriostatic agents, on the other hand, are those antimicrobial agents that cannot really kill your microbes, but they can only inhibit it from growing further to multiply. Okay, so it will uh, stop any metabolic processes. So dili gid mo tubo o dili pod mag-multiply imong microorganism. Now, we also come across the word germicide, which is another general term, okay, which would mean to kill germs. Okay? So, may iba't ibang klase yun. So, bactericide, pangpatay ng bacteria, virucide, pang-destroy ng viruses, fungicide, pang-destroy ng fungi, and sporicide. Pwedeng two-way yun, no? Pwedeng sporicide dahil it can destroy endospores of bacteria or it could destroy yung spores ng fungi. Okay? Now, moving forward, when we talk about microbial control, we have to be familiar with the factors that may affect, no, the processes of killing microbes. And this would include the following. No? So these are the factors that would influence the effectiveness of antimicrobial treatment. Number one, the number of microbes that you have. So say, for example, you're going to wash a week's amount or a week's load of laundry. Tapos ang imo lang gamiton kay isa naka sachet sa sabon. That's unfair. Okay? So di na siya silbi. So there should be an equivalent volume of your disinfectant to use to make sure na mamatay ang imuhang microbes. Okay? Next, we have the type of microbes. So also you have to look into the whether or not your microbes to kill or remove are infectious, pathogenic, or non-pathogenic. So you should be able to match it with an equally strong antimicrobial, not to kill your pathogenic organism. Okay, of course, and environmental influences. Now, this would refer to the type of water, the amount of water that you use, diba? Most of the time, we use a dilution of your bleach to clean materials. Okay? So, kay nagtipid ka, imong gigamit kay sa kabalde nga tubig, pag human, isa lang ka cup sa bleach ang imong gigamit, di na siya pwede. Okay? The water, what else? Baka naman may temperature na dapat ay i-follow. So you'll have to be able to read, no, yung manufacturer's details about how to use the product. Okay? So baka naman kailangan mainit na tubig, baka kailangan tap water lang, buot-buot ka. Okay, what else? Say for example, you're going to clean yung materials that would contain vomitus or phlegm or lipids, okay, they may mask your organisms. Okay? So, naglimpyo na ka, pero pwede sila namatay. Okay? So, the advice usually is, or whatever, the recommended process would be, if you have spills of vomitus, no, naisuka, so imo na siyang tabunan of tissue, and then pour over, no, enough volume of disinfectant to soak your tissue to ensure na mabasa po itong imuhang tawag niyan, nung vomitus, no? Or plema ba yan? Okay? So, dapat merong nilalagyan nga yun ng parang kinukordon yung area na may gano'n, no? So, around 15 minutes. Okay? And then, remove that. Okay? Tapos, second, ano na naman na disinfectant, no? Pangalawang linis para make sure na walang, ano. Tapos, yung tissue na kinuha, no? Yung may suka, tatapon mo siya in your yellow bin, no? Or in some cases, depende sa policy, in some hospitals, they would need to soak the rug or yung tissue in disinfectant, a container with disinfectant. Okay? And of course, fourth would be time of exposure. So kung sabi doon, 1 hour mo siyang ibabad, 1 hour mo siyang ibabad. No? Kung kailangan overnight, hindi overnight mo ibababad. Okay? Now, here is a list of your resistant microorganisms. So top of the list is your prions. And at the end, you have your viruses with lipid envelopes. Now, why is prion at the top of the list? Okay, prions are actually remnants of viruses. They're not even viruses. Now, they're just a part of what is supposed to be a virus. However, no, they are just basically proteins, but still, they are infectious. Okay, they are still able to cause other proteins to mimic their abnormal forms, hence producing many abnormal proteins. Okay? mahirap siyang hanapan ng antimicrobial agent, especially because of that characteristics, just a protein, okay? So you have to be able to match it with an antimicrobial that is strong enough to destroy the proteins that make up the prions. Viruses, on the other hand, with lipid envelopes, are quite easy to destroy because lipids is really sensitive to a lot of chemicals that we use as disinfectant, even alcohol. Okay? So, madamage ang envelope niya, adi ma-expose na yung kanyang capsid. So, pag na-expose yung capsid, madali na lang masira yun. And ma-expose yung kanyang genetic material. Ayan. Okay? So, again, no, nandito tayo sa ating physical methods, no, of microbial control. So physical methods are those that we can actually, expose our materials to be disinfected too. Okay, so this would include heat, low temperature. What else? Low temperature. You have the ano pa ba dun? Filtration, desiccation, radiation, yun yung iba't ibang klase ng physical methods that we can use to really attack our microbes with. Okay? So heat is the first and the most common. So heat usually destroys or kills microorganisms by denaturing their enzymes. And so pag na-denaturing enzymes, nadadamage din yung mga proteins na kailangan i-produce pag nawala yung enzymes. So when we're talking about heat, no, you'd have to be familiar with these three terms, no, yung thermal death point, thermal death time, and decimal reduction time. Thermal death point is the lowest temperature at which all the microbes in the liquid suspension will be killed in 10 minutes. So again, death point, lowest temperature that can kill microbes in 10 minutes. Okay? So, see, thermal death time naman, that's the minimum length of time that can kill a suspension of microbes at a given temperature. Okay? And then see decimal reduction time, the time in minutes at which 90% of your bacteria will be killed at a given temperature. So ito yung madalas na ginagamit or ina-apply na method of microbial control ng mga canning industry. Okay, so kaya yung mga sardinas natin, corned beef natin dyan, pwedeng-pwede na natin kainin na hindi pa naluluto dahil naman na-remove na yung microbe. So mo lang kainin na expired na. Okay. So under heat, we have moist and dry heat. No? So under moist heat, this would make use of water. Okay? So boiling ang pinaka-common, di ba? So boiling, exposing your microbes at 100 degrees Celsius or a little bit higher. So it could again kill, no, your microbes. Okay? So ito yung ginagamit natin na panglinis ng ating mga beberon, di ba yan yun? Okay? So moist heat can kill microorganisms by, of course, coagulating their proteins. Okay? It's actually very effective compared to dry heat. Okay? So boiling, again, is able to kill vegetative forms of bacterial pathogens, almost all viruses, masira siya, no? And fungi, as well as their spores, pwede nang mamatay as long as na-expose sila for 10 minutes. Okay, endospores and then some viruses may not be totally destroyed, no, by boiling tapos 10 minutes lang. Minsan kailangan ng mahabaang boiling. Okay, pero yung mga regular pathogens natin can be killed by brief boiling. Okay? Si hepatitis virus, kailangan medyo mahaba ang boiling, mga 30 minutes or more. Si endospores naman, ay, dili jud pwede ani. So, kailangan mas mataas than 100 degrees Celsius, like probably your autoclaving. Okay? So, sa autoclaving naman, eto water din na kailangan natin, no? So, kasi gagamitin natin siya as or in the form of steam. So, autoclaving is steam under pressure, no? We use 121 degrees Celsius and pressurize the organisms at 15 pounds per square inch. So, this is slightly higher than the atmospheric pressure, which is around 14 psi lang. But the difference between 14 and 15, 1 point lang, right? It's enough to kill your microbes. Feeling na natin, tiris na sila sa 1 point the difference. No? Tapos 15 minutes pa. Okay? Now, it's important for us, especially if you use the autoclave every day, no? And several times in a day. We need to make sure na we practice preventive maintenance para hindi siya nasisira, no? At saka talagang it is able to maintain its ability to kill microbes. Okay, so we should be able to check its function by using kilit ampules. Okay, kilit ampules are, ito yun, no? Nasa ampules. These are biological indicators. They contain Bacillus thermophilus, no, the spores of Bacillus thermophilus, to check whether or not your autoclave is working. Okay, so pinapasok natin purple. Okay, dapat lumabas yan purple pa rin, no, to mean your autoclave is working. Okay, however, if after autoclaving, no, nainitan yung ampule, naging yellow, ibig sabihin nun may microbe buhay. Okay? Binuhay mo tuloy yung spores, yung endospores. Okay? So a positive sign for autoclaves that are not really working or that would require autoclaving is nagiging yellow yung ating uh, kilit ampules. Okay. Now, pasteurization is another method employing moist heat, pero this one makes use of lesser temperatures. Okay. So we know for a fact that this one was established by Louis Pasteur, and it is created to sterilize very delicate material like beverages, food, dairy products. Okay. Now, there are three types of pasteurization method. They have the classical method, which would expose your materials to 63 degrees Celsius, pero for 30 minutes. So medyo mahaba. And then we also have the high temperature short time holding or short time method, which would expose your materials to 72 degrees Celsius, but 15 seconds, napaka-minimal. Okay? And then the best one would be ultra high temperature method. So ito yung ginagamit niya sa mga milk products. So exposing materials, excuse me, to 140 degrees Celsius, pero 30 seconds lang. Parang pumikit ka lang tapos na-sterilize na siya. So this method is commonly used in milk products. Okay? So commonly used in milk products natin. Ayan. So dito naman tayo sa dry heat. No? So for dry heat, this would kill the microbes by oxidation. So ito walang water, ha? So init lang talaga. Examples nito is direct flaming, no? Like those that we use when we sterilize our loops, our inoculating needles, and or the mouths of our flasks and tubes. And of course, pwede rin ito, di ba? So yung ating mga electric na uh, electric incinerators for your loops. We also have yung incinerator na ganito, no? Kaya lang, this one is not pwede in the Philippines because meron tayong Clean Air Act. So, bawal. Naka-pollute daw og hangin na to diri. Okay. Pero in the lab, this one is also very common, no? Your hot air oven. We use this to sterilize your plates, your glasswares, no? Your slides, also yung mga metalwares natin, like the loops, pwede rin naman siyang i-sterilize dito. So usually 160 degrees Celsius for 2 hours or 180 degrees Celsius for an hour pag nag-sterilize using your hot air oven. Okay, so aside sa heat, we also have filtration. So second method, no, filtration, HEPA filters, gaya ng mga nasa biosafety cabinets natin. Okay, ito yung itsura ng HEPA filter natin. And we can also make use of membrane pore filters. This one we used actually to remove solid materials, infectious agents from our liquid samples, like your body fluids, pleural fluids, blood, urine, yan. So pwede yan. Even viruses, large viruses can be filtered kasi meron tayo ng 0.01 micrometer. Yung mga pox virus, pwede sila dyan. Okay. Yan. Third method, we have the low temperature. So this one, especially refrigeration, it is microbiostatic. So it can only control growth and multiplication of your microbes. Okay? Pero pag bumalik sa tamang temperature, tubo na po sila dahil it's not really bactericidal. Okay? Freezing can be bactericidal, especially in slow freezing, because hinahinay na ma-form ang ice crystals, which could pierce your bacteria. or parasites, for example. Ang flash freezing, murag pareha lang po siya sa refrigeration. Okay? Siyempre, desiccation, no? So, remember, this would involve removal of water. Water is a very important, it's an indispensable requirement for growth, right? So, kung walay tubig, walay growth, walay reproduction, some, like those na may mga endospores, they can survive. Pero for those na walang endospores, mamatay yun. Um, like your Neisseria gonorrhoeae, it can only survive for 1 hour. Your TB bacillus can survive several months kasi nga naka-encase sila in the phlegm. Plus, unusual ang kanilang cell wall. Di ba ang cell wall nila nandun yung mycolic acid, which renders their cell walls waxy and so resistant yun sa labas ng tubig. It is able to keep water intact, no? Except kung medyo matagal na siya masyado naging desiccated, okay? Na viruses, syempre, walang ano yan, no? Hindi tumatalab yung desiccation for viruses as they are a cellular, okay? And of course, Clostridium and Bacillus species, they may survive several decades because they are able to produce endospores, okay? Then we have osmotic pressure. So, osmotic pressure, okay, lalo na pag nasa hypertonic environment, will promote plasmolysis. No? It will cause the organism to shrink. Okay? Ang cell membrane niya will shrink away from the cell wall, inhibiting growth and reproduction. No? Kasi walang communication between the two linings. Okay? Pag hypotonic naman, pumapasok ang tubig, it would swell. Again, no, the cell would swell. Again, it would affect growth and reproduction. Okay? Pero, yung mga organisms natin na exposed na, no, sa skin, kung saan medyo mataas ang osmotic pressure, like your staphylococcus, they're not really as affected, no, by osmotic pressure. And syempre, we have radiation. Okay? So sa radiation, we have ionizing and non-ionizing radiation. Okay? So this one, sa ionizing, medyo shorter ang kanyang wavelengths, meaning yung distances between yung beams is shorter. So mas dali siya damage sa genetic material. Okay? So a short exposure, no, to ionizing radiation may be detrimental, okay? So, especially pag yung matataas na masyado. So gamma rays, X-rays, electron beams, UV rays, especially yung mga 150 nanometers ang wavelength, they can cause DNA mutations and they can produce yung mga free radicals like peroxides, which are deadly for your microbes and at the same time damaging po sa ato ang tissues. No? So this is effective for pharmaceuticals and medical supplies. Okay? And then we have yung non-ionizing radiation. Okay? So ito yung kailangan ng long-term exposure. Kasi nga, medyo malayo ang kanyang wavelength, no? Larger than 1 nanometer. So ito yung kailangan ng matagalang exposure para magkaroon siya nung mutations. Okay? Magpa-pile up lang yung mutation pag lagi-lagi kang na-expose. Gaya ng, di ba kaya nga hindi pwede tayo magpa-x-ray ng ma-expose na sa ating mga radio waves parati. Dapat hindi pwedeng itinatabi yung cellphone or naka-open yung laptop mo tapos natutulog ka ng matagal because it can cause cellular mutations, no? Nagdi-DNA mutations tayo, lalo na pag matagal na exposure, no? Mag-accumulate yung tawag niyan, yung damaging effect ng non-ionizing radiation. Okay? So moving on to chemical methods of microbial control. Okay? So sa chemical methods, syempre, ang gamit natin, chemicals, no? First, we have phenols and phenolics. Ang phenols, ito yung inadvocate ni Joseph Lister, di ba? Nagumamit ng phenol para panglinis ng mga ginagamit for surgery. Initially, they even used this to clean wounds. However, masyado siyang matapang, yung amoy niya. And then, halang po sa skin, no? It's a skin irritant. So, eventually, na-change nila yon. Gumawa sila ng phenolics. Okay? So these are chemical derivatives of phenol. Okay? Like your cresols, no? So under cresols, you have your O-phenylphenol, like your Lysol, and then you have your bisphenols, like the triclosan. Ang triclosan is an active ingredient in the antimicrobial soap na safe, no, not safeguard, Lifebuoy. Okay? So, ang advantages ng phenols and phenolics is that it's stable. So, pwede siyang mag-last, no? Mag-persist for a long time. And then, it can remain active even when combined with other compounds. Okay? And then we have halogens. So, ang halogens can be used alone or it is effective in a compound, no? Together. So very, very familiar itong dalawa sa atin, like iodine, no? So under iodine, you have tincture of iodine and you have iodophores. So iodophores like betadine and iodine. So alam natin, these are antiseptic solutions, right? And then chlorine. So chlorine is effective in disinfecting drinking water, sewage, of course, your swimming pool. So chlorine will, when combined with water, will produce hydrogen molecules and chloride plus hypochlorite. Okay, so yun yung ating pinaka-active ingredient actually. Then alcohols can also kill microbes, no? Yun nga yung gamit-gamit natin araw-araw, right? So, mayroon tayong ethanol and isopropanol, methanol even. However, we rarely use that one for disinfection. We use that for fixation instead. Even then, hindi din tayo masyado gumagamit kasi it was known na toxic po ni, no, si methanol. So instead, for the usual disinfection that we do, we can use isopropanol or ethanol. Pero between the two, your isopropanol is a better disinfectant. Okay? And then heavy metals, ayan. So heavy metals are also observed to have oligodynamic action. Meron, pero konti lang, like this, no? So that's the coin, and you can see a zone of inhibition, very small, pero naa. Okay, so there are several types of heavy metals that can use for disinfection, like your silver. Okay, so like in the form of 1% silver nitrate, so combination. And you have mercury in the form of merthiolate or mercurochrome. No? Ngayon, na ginagamit na natin na uh, pang manicure, pedicure, para pink-pink. Okay, and then of course, copper sulfate, your selenium, no? Selenium sulfide, which is actually used to destroy fungal spores. And then zinc, no? In the form of zinc chloride or zinc oxide. Okay. Siyempre, we can also make use of your detergents, no? Or those that belong under quaternary ammonium compounds or quats. So these are surface active agents, which are proven to be more effective against gram-positive organisms than gram-negative organisms. And yung ating mga aldehydes, okay? So aldehydes like your formaldehyde and your glutaraldehyde are also very effective fixing agents, okay? So we use them in sopa. Ang formaldehyde gas can be used as a disinfectant. And then yung kanyang liquid form is yung ginagamit natin for fixation, no, of tissues. So this has a really strong odor. Masakit sa mata, masakit din sa ilong. Okay? Si glutaraldehyde is more effective as a disinfectant compared to formaldehyde gas. No? So it's widely used as a sterilizing agent, especially for hospital instruments. And then meron tayo ng gaseous sterilizers like ethylene oxide, pero which is actually quite effective. We use this to disinfect your hospital linens, yan. Pero you'll need long exposure. So 4 to 18 hours of exposure to ethylene oxide to ensure na mamatay ang mga microbes. Okay? And then, of course, we have the peroxides. Peroxides are oxidizing agents. So they are able to disrupt membranes, no, and proteins. Example of this is the ozone, which can be produced when oxygen is exposed to electricity or ultraviolet light. Okay? So it's the highly reactive form of oxygen. It acts like chlorine, pero mas stable lang yun si chlorine and less expensive compared kay ozone. And then we have your hydrogen peroxide, which is able to break down, no, yung which is very effective, no, to kill no, microorganisms. So it's also used as an antiseptic. Dati, I remember when I was a child, mas common itong hydrogen peroxide compared sa betadine. Okay? So it may be sporicidal also. Ang benzoyl peroxide, we know this as an acne medication. And yung ating peracetic acid is a sterilant. It's a very effective liquid sporicide. Okay?